Hello, boys and girls. This is Mr. So. Welcome back for another lesson of biology. Today we are starting a new chapter, chapter 28, about the biotechnology. And this is lesson 34 about the recombinant DNA technology. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. What is biotechnology? It is the use of organisms or biological systems or processes in producing goods or producing services, such as we put a human insulin gene into a bacteria DNA. And then afterwards, the bacteria will use the human insulin gene to produce human insulin, so which is a human protein. This is biotechnology. Besides producing human insulin using bacteria, recombinant DNA technology can be used in fruits or vegetables as well. On the left, you have a genetically modified tomato, and on the right, you have a natural tomato. They have both been put on table for more than a week, while with the left, GM tomato still being freshy and juicy, but the right, natural tomato looks so old and ripened 成熟. Because on the genetically modified tomato, the GM tomato, we have put a gene to slow down the ripening process, 成熟嘅過程. So these, after modification, we call this organism a genetically modified organisms. For the production of GMO, it is actually an application for the genetic engineering. And this process helps to change the genetic makeup of an organism. So what is the recombinant DNA technology, DNA 重組技術? It is a technique used in genetic engineering. It allows the scientists to introduce a new characteristics or a phenotype into an organism, such as we put the human insulin gene into the bacteria and requires the bacteria to use the human insulin gene to produce human insulin protein. The step for DNA recombinant technology is shown below. First, you have to obtain the DNA fragment containing the gene of interest. That means, for example, this time you would like to use the insulin gene that you're interested in using. This is the insulin gene maybe. We take it out from a human body. And after that, with the gene of interest, we can do a little bit of modification. Step 2, you would like to obtain the DNA from a bacteria as well. Now, inside the bacteria, there are two types of DNA. The first type of DNA, they are highly coiled, very, very big inside the bacteria. We call this the chromosomal DNA. They are very, very big in nature, very, very long, highly coiled. And then another type of DNA, they are rather small and they are in a circle shaped, and we call this the extra chromosomal DNA because it is outside the original chromosomal DNA, so we call that the extra chromosomal DNA, and it is like a small ring, and we give it a special name. We call it a plasmid. This plasmid is a very good DNA carrier, so it can be used as a vector 載體 to carry later on the human insulin gene into this plasmid. After we obtain the DNA fragment, that means the gene of interest, and the plasmid, we can use a restriction enzyme to cut the plasmid and the gene of interest. Very importantly, we are going to use the same restriction enzyme to cut the both edges of the gene of interest, and also same restriction enzyme to cut on the plasmid. The restriction enzyme cuts the gene of interest and the plasmid opened with sticky ends on the two sides. After production of the sticky end, which looks like a ladder or just like a puzzle, it will be easier for the insulin gene, the gene of interest, to bind with the opened plasmid. And these sticky ends, if you look into the details of it, it is actually having complementary base pairing. So it allows a very complementary and strong sticking between the bases. After the combination of the gene of interest, such as the insulin gene, into the opened plasmid, now another enzyme is coming out for help. We call this the DNA ligase. It's an enzyme which helps to join the DNA fragment, that means the gene of interest, and the plasmid together, and helps to make sure the joining is very secure and strong. The recombinant plasmid, which now carries the insulin gene, a new human insulin gene, will now be introduced back into the host cells, such as the bacteria. Put it back, and if the bacteria contains the human insulin gene, it will produce the human insulin, and which is a protein. And this gives the bacteria a new character. And the bacteria now giving human insulin is also good. It provides a mass production of insulin to all the diabetic patients in the human population. Here comes a question. So after the human insulin gene is inserted into the bacterial plasmid and put it back into the bacteria host cells, is the insulin produced a bacterial protein or a human protein? Can you explain why? Give you 5 seconds to think. The answer is the human insulin gene inserted into the bacterial plasmid, put it back into the bacteria. The protein produced should be the human protein because it's a human insulin gene. No matter in what organisms with the human insulin gene, it must produce the human insulin protein. And the explanation is because of the property of the genetic code or the triplet code, they are universal. So it always codes for the same amino acid. No matter in what organisms, it will always express to give the gene and give the protein. For example, if this time it is a human insulin gene, it will always give the human insulin protein. The following case shows you another example of DNA recombinant technology using the gene for human growth hormone. Step 1. To obtain the gene of interest, that is, to obtain the gene for human growth hormone from a human cell. Step 2. To obtain the vector plasmid from a bacterial cell. Step 3. Use the same restriction enzyme to cut on the gene for human growth hormone and to cut on the plasmid to give both sticky ends on the human growth hormone gene and the plasmid. After formation of the sticky ends on the human growth hormone gene and the plasmid, DNA recombination occurs. Step 4. DNA ligase, an enzyme, is used to join the gene for human growth hormone with the bacterial plasmid. After joining with the help of DNA ligase, a recombinant plasmid is formed. The recombinant plasmid is now transferred and inserted to the host cell, such as the bacterial cell. The bacterial cell now containing the gene for human growth hormone can have the human growth hormone production. For your information only, scientists have developed a more advanced technology to help inserting the recombinant plasmid or the recombinant DNA into the host cell, such as in the following diagram, you see this is a gene gun, 基因槍, so it helps to shoot the recombinant plasmid or recombinant DNA into the host cell with a faster and more efficient way. Please go to your textbook page 9 to finish the MCQ over there. That's it for the lesson of recombinant DNA technology. I will see you in the next lesson. Bye bye.